To everybody, myself, Dr. Jyotirmay Biswas, who is working on, I'm working on uveitis and ophthalmic pathology at Chennai at Shankar Nethralaya. I welcome on behalf of Society of Inflammation Research for this virtual bi-monthly lecture series of immunology and inflammation. As you know, immunology is quite important, inflammation is quite important in the basics of all diseases, infectious or non-infectious. And our society is relatively new, almost started in 2017. We have got several members. We meet annually. Annual meeting used to be there earlier physically, now is going to be virtual. We have a journal of our own, and we are launching one book on inflammation, which will be coming soon in 2 volumes. We have got several other activities. We have one of them is this bi-monthly lecture series, which started from March, and we have designed for lectures till December. And due to the COVID pandemic, we have to have this kind of virtual lecture series, and we have got eminent speakers from the various parts of the country, even from abroad. So the society is comprises of several medical specialists, like various branches of medicine, like rheumatology, ophthalmology, diabetology, etc., as also from basic sciences. If you are interested in the inflammation, you can join this immunology and inflammation. You can join the Society of Inflammation Research. Our website is there. You can get the membership form. Today is a very interesting topic, innate response of the inflammation, and we'll be having 3 speakers. I request Dr. A.S. Kamalnathan and Dr. Mohan Wani to be the moderator of this today's second series of bi-monthly lecture on immunology and inflammation. This is innate response of inflammation. Over to them.
So good evening, everyone. I am Dr. Kamal here from VIT, that is Vellore Institute of Technology, Vellore, and I'll be co-chairing or moderating along with Dr. Mohan Wani for the next few minutes. So let me introduction slide of Dr. Himanshu Kumar. Can this be blown up? No, full screen? Yeah, yes. Thank you. Okay, let me introduce you as the speaker of the first event. So this is Dr. Himanshu Kumar. He is an associate professor working in Department of Biological Sciences at ISR, Bhopal. And his basic research interest is on innate immune biology, where he focuses on the transcription and translation regulations of antiviral innate immune responses, not only for specifically to the flu, for even other viral infections. So he obtained his bachelor's and master's degree from Lucknow University. Following that, he obtained his PhD from Japan, Osaka University, Japan in 2008, and was awarded as an excellent PhD student of a medical school. Following that, he got a postdoctoral fellowship from the Japan government, and where he continued for 2 years. After that, he moved back to ISR, Bhopal in 2010 as an assistant professor. Currently, he's serving as an associate professor, and he has a lot of credits on his list. So let me cut short here and allow him to talk about the topic of the day. Okay, thank you. Dr. Himanshu, can you share your slides?
Shall I start?
Yeah, please go ahead.
Yeah. So first of all, I am very happy that I am a part of this lecture series, and I will initiate this or I will try to give the momentum to this lecture series through my first talk, and I think I will motivate you sufficiently so that one should look at the inflammation. If you look at any disease, any problem, everything is based on inflammation. And today I will try to convince. I'm not sharing today my research data. I'm just giving an overview, and in future, if required, I can talk about my basic research, which is I'm doing with various viruses. Okay. So very great introduction, my introduction, and I'm very much thankful for all.
Himanshu, your slides are not visible.
Yeah, share the slides, please.
Share the slides, please.
Okay. So you cannot see the slide.
No, not it. Iyengar, you have.
Now you are able to see.
Yeah,
No,
sure.
oh, yeah.
No, you are right.
It's fine now.
Yes.
Yes.
Please go ahead.
Okay. Sorry for this. I don't know. But initially it was visible. Anyway, let's move on. So the title of my talk is, I will talk about the innate immunity and molecular pathogen recognition. I made this title little simpler, what I was given, because it will be a bit complicated if I put that title, and this will be easy to follow, and I will deliver what you all are expected from me. So in this talk, I will talk about most important thing is the inflammation. The inflammation is, it's a variety of or root of most of disease or problem. And I will talk about the basics of inflammation, some concepts of inflammation, and its outcome in terms of there are variety or spectrum of inflammation, and there is a variety of outcomes. So I will discuss about the outcome, and then I will very strongly link with innate immunity. And innate immunity, previously it was thought that this is a non-specific response of host, but now we know it is not non-specific. It has a broad specificity, and this concept was initiated by the Charlie Janway, and subsequently it was very well worked out. So I will talk about that component in more detail, and that component we call it as a pattern recognition receptor. So let me start with inflammation. So these are the cardinal of inflammation, and this concept of inflammation was first given by the Roman physician. This name is a bit complicated, but I will try to speak. I learned the pronunciation, honestly. Cornelius Celsus in 1st century AD, and he gave a concept of inflammation. And what is that? This is a rubor et tumor com caloro et dolor. That means that redness and swelling with heat and pain. So this is the 4 points which is associated with the inflammation. But after this, probably you are very well aware about one name known as Emil Metchnikoff, who discovered the phagocytosis. So he also worked on inflammation, and he gave the concept of inflammation in positive sense. This inflammation is good or important for the clearance of pathogen. Okay, and in contrast, another scientist, Rudolf Virchow, he gave this inflammation is only associated with some pathogenesis or disease or pathological effect of inflammation was well worked out by the Virchow. And he gave one more cardinal, that is disturbance of function, that is factiosa. And if you see this image, I got it from the internet, and I thought it is very nice. So this inflammation here, it is depicted in this cartoon, that is heat, redness, swelling, pain, and loss of function. So these are the key cardinals of inflammation. And it is not necessary always that all these things will be there when inflammation is there. maybe sometime 1 or 2 factors will be there, or maybe 3 or 4 or something like that. It's not necessary that all factors of inflammation will be there. And when we say that, okay, this is a heat, redness, swelling, pain, and loss of function. So what is the cellular basis of this heat, redness, swelling, pain, and loss of function? So here I will just give again a very simple concept. So when heat will be generated, so when there will be a vasodilation and there is an increase in blood flow, so that will cause some kind of heat. And when there will be an inflammation, then there will be a loss of permeability, there will be a loss of fluid from the vessels, and there will be increase in permeability, and that results to the swelling and redness. And there will be extravasation. So there will be a factors released by immune cell that will cause the extravasation. Extravasation is the movement of immune cell across the blood vessel. Okay. So that will give the tenderness, and it may also give the pain, and all these things results to the pain. Okay. So this is a kind of physiological impact of inflammation. This is a very generalized thing. This may be a complicated, that depends on the situation. So let's look at what are the causes of inflammation. So generally we think that there is only single cause, but today I will give the concept that there are 4 major causes of inflammation. You can categorize all kind of inflammation in these 4. One could be the microbial infection, any microbial insult to the host that will cause the inflammation. It could be a bacteria, virus, or any microbial. Or it could be unfavorable living and non-living entities. Living and non-living entities means it's not always the pathogen. There could be some living pollen that can go into the host system, and that may lead to the inflammation. Non-living, okay, there are so many things like dust, now asbestos, silica. There are so many things, pollutant. All those things can also result to the inflammation. Inflammation can be also induced by the tissue injuries. So tissue injuries, for example, if you have a very clean hand and you have a very clean blade, then if you make a cut on your hand, then that results to the inflammation. That will also, we call it as a sterile inflammation. And if I have opportunity, I will talk more about that. And then inflammation can be also due to the loss of homeostasis by intrinsic and extrinsic factors. Intrinsic factors means, for example, a very simple example, we have all normal values or range of values. Okay, let's take the example of glucose. So if the glucose is extremely high, then that also results to the inflammation. There will be some inflammation in tissue, and that results to the other complication. Okay. So what is the impact of all these kind of inflammation? So if microbe is inducing inflammation, then that may result to the pathogen clearance. But this should be in some particular range. Okay. And the pathological outcome will be if this inflammation is uncontrollable, then that lead to the pathology or immunopathology, microbe induced pathology, and that could be a sepsis, tissue damage, and that may also result to the autoimmune disease. Now you are in current pandemic situation, you can understand that there is some term people quite frequently use. This is known as cytokine storm. That is a too much inflammation. Okay. And I will tell how too much inflammation can be amplified in the host in later slide. Another is that if there will be unfavorable living and non-living entity challenge, then there will be a clearance of living and non-living entity. So our system will try to clear it. But if it is not able to clear, then that may result to the allergy or anaphylaxis. And if the tissue-induced inflammation will result to the tissue repair. And if this inflammation is not controlled, then that may lead to the tissue fibrosis, which may result to the oncogenesis. Okay. And another is that if there will be a loss of homeostasis, then our body or our system will try to adapt with this stress, or try to restore or bring it back to the homeostatic position situation. But if it is not controlled, then that will lead to the homeostasis associated disease. So this table will give almost all kind of information about the inflammation. Okay, so what is the correlation between inflammation, homeostasis, and disease? This is a simple pendulum. You can see that there is a bob and it is moving. So there is something we call it as a homeostatic range. Again, I can give you the example of, say, blood sugar or any factor. So there is a range. If it is going below, then that is also not good, and if it is going above, then that may result to the inflammation. And any perturbation means it's an intrinsic or extrinsic perturbation. For example, insulin, if sufficient amount of insulin is not there, then that will kick the glucose values in out of range, and that we can call it as a perturbation. Or whatever the cause of inflammation, say microbial infection, that will kick the inflammatory cytokine production in higher amounts. So that is also known as perturbation. Okay, so any perturbation may result to the physiological inflammation. Physiological inflammation means that will be initially it will be good for the host in order to repair or in order to recruit the immune cell if it is an infection, and then it will try to clear by the that normal level of inflammation, and that we call it as a physiological inflammation. But there could be sorry, this is a pathological inflammation. The last, it's not physiological inflammation. So this is a typo. So this pathological inflammation result to the complicated diseases. And this pathological inflammation can be induced by persistence of inflammatory inducer. For example, if you take the coronavirus, so if there is a too much virus replication, then there will be a too much cytokine production, inflammatory cytokine, type I interferon, and all those things. Okay, so that will cause the persistent inflammation. It can be also due to that the inflammation which is induced by the inducer is not cleared by the system. Okay, system is our body. If the inflammatory substances will be remain there, and it is not cleared efficiently by our system, then that will also lead to the pathological condition. Okay. Another reason is that this inducer basically cause some kind of damage, and those damaged material can lead to the further amplification of inflammation. Means it's a kind of synergistic effect. For example, if virus is replicating too much in the cell, and then that is causing the cell death. Okay, not only the apoptosis. Besides apoptosis, there is a death of cells. Okay, and those dead cells will release the DAMs, danger associated or damage associated molecular pattern. So those danger associated molecular pattern, again, they will induce the inflammatory pathway, and then there will be too much cytokine and too much inflammatory things will be there, and that will cause, for example, in case of several viruses, there is a cytokine storm. So these are the pathological inflammation. Okay. So now I will move. I just gave a quite broad perspective about the inflammation. Now I will talk about the innate immunity. So innate immunity has a several component. It's a very simple. It's like a physical barrier. There is a biochemical barrier. There will be a microbial barriers, and there will be a soluble factors, which is providing the defense against the infection or any foreign entity. And this is consist of antimicrobial peptide. You know probably very well. There is a CRP. These days, doctor suggests to check the CRP, C-reactive protein, mannose binding protein, and there are variety of protein. And particularly, I would like to bring your attention to the complements. The complements are very important. They are the family of protein, and these family of protein basically induce various kind of physiological effect. Complement can cause the lysis of target cell if the complement will be activated. it can also induce the inflammation. Probably you remember that the smaller component of complement can induce the inflammation. Okay, and complements can also induce the opsonization. If the microbe is coated by the complement, particularly C3b, then it will be readily phagocytosed. So these are the soluble factors of innate immunity. And there are the cellular component, and the cellular components are almost all cells. It includes immune and non-immune cell, and particularly like macrophages and DCs. So these cellular component, they have some special kind of receptor which is conceptualized by the Charlie Janway, and later on this was this was discovered by the Ruslan Majitov, and then Bruce Butler, Shizuo Akira, and all these guys have cracked out all the pathways and everything. Okay, so now just I will quickly go through this physical and biochemical barrier. So this could be a cilia. Ciliary movement will keep the foreign particle or bacteria or virus, whatever, out of the system. The mucus, which is secreted over the respiratory tract, that will also help in clearance of pathogen. Cuff reflex. So this cuff reflex is also important to, if something goes inside, then this cuff reflex will also kick it out of the system. Mucus secreted again, the acid in gastric reflex. Intestinal enzymes. There are so many enzymes or antimicrobial peptide which is present in this in intestine. Then microflora. There are so many good bacteria. We all know about this. And the skin and the sweat and sebaceous secretion. So this keep a pH, maintain the pH, which is not good for pathogenic microbe. Okay. So what are the key players? So key players are neutrophil, eosinophil, basophil, and mast cell and natural killer cell. I just put few cells, not all. And these cells are basically playing a very important role in first line of defense. And adaptive immune cells, you all know that B lymphocyte and T lymphocyte, they are the key player. So these innate and adaptive immunity is not two separate entity. It is joined by these key cells. And if you look at the humoral component, then complements are the one of humoral factor which joins innate as well as adaptive immunity. Probably you know that classical complement pathway that requires an antibody. Okay. So similarly, this macrophages and DCs are the junctional cell, which plays a important role for innate immunity as well as adaptive immunity. Okay. So now let's look at how this innate immune, how this cellular component works. So I have already explained about the humoral component. The cellular component I have told it is all non-immune cell and immune cell. So these cells basically expresses a family of receptor. There are several families of receptor, which we call it as a pattern recognition receptor. And this pattern recognition receptor, they basically senses the signature molecules of pathogen, and those molecules are essential for their replication and for their cellular processes. And those molecules we call it as a pathogen associated molecular pattern. So PRRs, which is expressed on our cell, this senses the PAMPs or pathogen associated molecular pattern, and this initiates the inflammation. Inflammation, this also activate the innate immune response. So when there will be some inflammatory cytokine in a microenvironment, okay, then if some phagocytic cells will be there, then that will be activated, and that activated cell will be far more capable to phagocytose. And this can also, when this sensing of PAMP will be taken place by the PRRs, then this will also induce the type 1 and type 3 interferons. So these type 1 and type 3 interferons are playing very important role in virus infection. Okay, so this is all needed. And most of things in general, we basically rely on the innate immunity. Everything is cleared by the by innate immune system. But if it is not cleared by the innate immune system, then all these factors, basically create a base for the development of appropriate pathogen-specific innate, adaptive immunity, which is basically triggered by the B and T lymphocyte. Okay, so this is a basic scheme and how it is linking with the adaptive immunity. Now I will quickly move on this pattern recognition receptor. So this is a little complex figure, but it is far more complex. Believe me, it is far more complex. Here I am presenting very simple schematic. So this has a one family of pattern recognition receptor, which we call it as a toll-like receptor. And I will talk more about the toll-like receptor in later slide. And you can see this is a very beautiful system. This toll-like receptor basically, so this toll-like receptor basically present on a cell membrane. So if something comes over the, or if something present outside the cell, then this pattern recognition receptor can take care. But you know, several microbes like bacteria as well as viruses, they undergo the receptor mediated endocytosis or some kind of endocytosis. So in endosomes also we have a pattern recognition receptor which can sense these microbes, and then it will induce appropriate innate immune response through NF-kappa B, which will induce the pro-inflammatory cytokine, and that can also induce the type I and type III interferon. So this toll-like receptors are very well placed in order to check the microbial challenges. Okay, we have a pattern recognition inside the cell also in a soluble form, that is, we call it as a Rigi-like receptor, and it has a 3-member. So I will talk more about these 3 family of pattern recognition receptor. Here just you can take a look that this will also induce the type I interferon and pro-inflammatory cytokine. There is another cytosolic sensor. This we call it as a NOD-like receptor, and then this also induces the mainly they induce the pro-inflammatory cytokine. They are not too much involved in type I interferon. And there are some DNA sensors also. Here I am not showing. So DNA sensors are present in the cytoplasm. And the adapter for this DNA sensors are this sting molecule. Here you can see there is a sting molecule. And this will induce the type I interferon and pro-inflammatory cytokine. There is some specific receptor, which is, we call it as a dactin-like receptor or CLRs. So they are basically senses the fungal signature, basically beta glucan and all those things. Okay. So today in subsequent talk, I will talk about the toll-like receptor, Rigi-like receptor, and NOD-like receptor very briefly. Okay. So what are the inflammatory mediators? That is very important. We are seeing there is induction of inflammatory signatures and type I interferon. So what are those inflammatory mediators? It could be a vasoactive amine like a histamine and serotonin. It could be ascanoids. You know very well ascanoid, which is basically synthesized from the arachidonic acid by the action of COX-1 and COX-2. Probably you remember cyclooxygenase 1 and cyclooxygenase 2, and this cyclooxygenase is basically inhibited by the several drugs, particularly aspirin. Okay, so ascanoids are also inducing the inflammation. Some peptides are also there, like brandycarb, and there are a lot of protein. You know very well. This includes the complement components, a smaller component, and very well qualified proteins, which we call it as a cytokine, like IL-1, beta, IL-6, TNF. So these are the inflammatory mediators. These induces the inflammation. What about the type I interferon? So type I interferon is basically a quite big family. There are 2 categories, interferon alpha and interferon beta. Interferon alpha, in human, there are 13 members, and single interferon beta is there, and they are basically inducing an antiviral state. And what is antiviral state? I will show you in next slide. And there are type I interferon inducible genes. So these interferon induces thousands of genes in order to create an antiviral state in the host. Okay, and this consists of IP10, rentis. There are so many. And there is a type 3 interferon. Not recently, but in recent past, this type 3 interferon was discussed, and this has been shown in several diseases, particularly in viral diseases. So there are three members, and we also call it as an interferon lambda. So how this inflammatory cytokine and type 1 interferon work, I will show you in a very simpler, extremely simpler way. So type 1 and type 3 interferon, basically it induces the apoptosis of virally infected cell in order to protect the healthy cells. Okay, so this will induce the apoptosis. This will make the healthy cells resistant from the virus infection. Okay, this is very important. Okay, and then you know that viruses hijack the host cellular machinery, and then they make their own copies. Okay, so type 1 and type 3 interferon, they basically suppresses the cellular machinery so that they cannot use it for making more copies. Okay, and they can also prepare and activate the professional immune cell, what I just now told, that B and T lymphocyte, okay, for the development of virus specific immunity. You know, there are so many kinds of T cells, particularly Th cells, okay, Th1, Th2, Th17, Th9, Treg, and so on. Okay, so basically this will prepare the type 1 and type 3 interferon, basically prepare the system in order to develop the virus specific immunity. What inflammatory cytokine is doing, this is basically an alert system. Okay, so they will alert the whole body in order to bring the immune cells at the site of infection, or if it is a systemic, then it will activate all immunity in order to clear this infection as soon as possible, immediately, or whatever. Okay, and this can be understood by, I'm talking about in context of virus because we work with viruses. So when there is a virus infection, our immunity, when immunity is not activated, our situation is something like that. Virus is quite strong, okay? But when we have, when the system is producing a lot of type 1 interferon or type 3 interferon, then our immunity is sufficient to fight with the viruses, okay? So this is very important. Okay, so this is the overall role of inflammatory cytokine and type 1 interferon. Okay, now I will talk about the pattern recognition receptor in little more detail. So I have already told that TLRs are basically present on basically they recognize bacteria, fungi, viruses on cell surface as well as endosome. The guy I told you that it is sensing, it's a cytosolic sensor and basically senses the RNA molecule. There is a DNA sensor. They also sense the DNA molecule. Okay, and RNA sensors are also involved in DNA sensing. In some, there are several literature. Okay, and there is a nod-like receptor which basically this is also intracellular sensor which recognizes wide range of PAMP, especially there is one molecule known as NLRP3. I will talk about that in later slide. That senses variety of living, non-living, everything they senses, and that induces the inflammatory cytokine, and it is also involved in activation of one multi-protein complex known as inflammasome, which is responsible for the production of this IL-1 family cytokines. Okay, IL-1 beta, basically it is involved in production of IL-1 beta. So toll-like receptor, I will give little detail. So this was originally discovered in the flies. It's it's an invertebrate, and there is some protein known as pencil, which is recognized by toll protein, and there will be a cascade of signaling, and that will induce the antimicrobial peptide. So here you can see this fly is heavily infected by the fungus. Okay? So over there, so this Julie Hoffman, his group, he find out this thing, and then later on, at the same time, not later on, at the same time, Charlie Janway and Ruslan Medjitov, they were also working, and they found out that there is a homologue of this toll protein. Okay, and this toll protein homologue is also playing a very important role in sensing some signature molecule of the pathogen, particularly LPS and peptidoglycan. This is a Nature paper which was published in 1997. Okay, And later on, Bruce Butler also showed this thing in the mice. He found out a mutation which is inactivated the TLR4, and then that hampers the innate immune response against the LPS challenge. So this field was started by many workers, and finally this work received the attention. And in 2011, the Nobel Prize was given to the Bruce Butler, Julie Hoffman, and Steinman. Steinman is not directly associated with TLR field, but he discovered the most important thing, the dendritic cells. Okay, so for that he got the Nobel Prize. Later on, several groups dissected this whole pathway, the toll-like receptor pathway, including the Professor Shizuo Akira in Osaka University. He made a huge number of knockout mice for all those signaling molecule, and he dissected all the mechanism. Okay, so how this toll-like TLRs are look like? So basically, these TLRs are having a leucine-rich repeat. Here you can see this ectodomain, this leucine-rich repeat, and there are TIR domain, which is similar to the IL-1 receptor and IL-18 receptor, and this domain is needed for the downstream signaling. Okay, and how the signaling is taking place, I will show you. And what these TLRs are sensing, I will quickly show you. Here you can see that these TLR1, 2, and TLR2, 6, and TLR4, they can senses the lipid and lipoprotein. Okay, and TLR5 and TLR11, they can sense the proteins, and TLR3, 7, 8, 9. So in case of human, in case of mouse, TLR8, we don't know the function, but capable to sense the molecules from pathogen. It could be a lipid, protein, and nucleic acid. So generally, this microbial entity has these signatures, okay, and this can be sensed by the TLR family protein. So TLR signaling is basically in we study in two major parts: mediated dependent signaling and TRIF-dependent signaling. Mediated is stand for this myeloid differentiation primary response gene 88. And when the mediated signaling is activated, then it will activate the NF-kappa B. And there is a TRIF-dependent signaling, which is mainly inducing the type I interferon beta, and TRIF is stand for this TIR containing adapter inducing interferon beta. Okay. So this mediated dependent signaling is predominant in macrophages, DCs, and plasmacytoid dendritic cells. So here there is one more kind of dendritic cell known as plasmacytoid dendritic cell. So plasmacytoid dendritic cells, or PDCs, plays a very important role in virus infection. They can produce ton amount of type I interferon. Okay, I will show you how it is doing. It is happening in the cell. And TRIF-dependent signaling is mainly taking place in the macrophages and DCs, and only two TLRs are involved in TRIF-dependent signaling. One is TLR3 and another is TLR4. So this is the mediated dependency. So this is a mediated is adapter, and this is used by all TLRs except TLR3. Here you can see TLR3 cannot be used by mediated. And this will activate. There are several kinds of IREC protein, okay, and TRAF6, and this will activate the IKK complex. You know, probably IKK alpha, beta, and gamma. So this makes a IKK complex, and this once IKK complex is activated, then this will phosphorylate the I-kappa B, which is present in bound form with NF-kappa B, and this will be, once it is phosphorylated, then it will be dislodged and it will be degraded through ubiquitination, and NF-kappa B will be transported into the nucleus, and then that will initiate the inflammatory cytokine synthesis. Okay. TRAF6 and this whole signalosome can also activate the MAP kinases, and that will also induce the inflammatory cytokine. In case of PDCs, this MyD88 can directly interact with IRF7. Please note that this IRF7 is a transcription factor which is required for the synthesis of type I interferon. So this transcription factor can directly interact with MyD88 in plasma cytoid dendritic cells, and then that will induce the huge amount of type I interferon in virus infection or during virus infection. Okay, so in that way, this is kind of short circuiting of the pathway in order to produce more amount of type I interferon in shorter duration. Okay. So now I will talk about the TRIF-dependent signaling. So this is basically, this TRIF is used by TLR3 and TLR4. And TLR3 directly interact with TRIF, but TLR4 needs another protein known as TRAM. Okay, I have not shown here. So this TRAM is an intermediate between TLR4 and TRIF. Okay, and then basically they can also activate NF-kappa B through MAP kinases and the pathway which I have explained to you. And they can also activate the kinases known as TBK1. This is a kinase which can phosphorylate the IRFs. Okay, IRF, there are so many kinds of IRF, IRF3, IRF7. So this will phosphorylate and activate this transcription factor, and then that will induce the type I interferon production. Okay. Now I will move to the RIG-I pathway. I will take a few more minutes. So RIG-I pathway, as I told you, this is a cytosolic sensor, and this cytosolic sensor basically play a very important role in sensing RNA viruses. So there are three molecules, RIG-I and MDF-I, and there is one more molecule known as LGP2. So LGP2 molecule, they don't have this CAR domain. Here you can see this is a CAR domain, and it is needed for the downstream signaling, okay, for production of type I interferon and pro-inflammatory cytokine. So LGP2 doesn't have this CAR domain. And initially we thought that this molecule doesn't induce this type I interferon and pro-inflammatory cytokine during virus infection. And in vitro study that also suggested same. However, when we made the knockout mice for LGP2, then we learned that this is enhancing the response. So now it sounds like that this LGP2 is acting above this molecule, okay, which enhances the responses. Okay. So this RIG-I and MDF-I have a different virus preferences. The bottom line is that this RIG-I basically senses the smaller pieces of RNA, and MDF-I can sense the larger pieces of RNA molecule, and it could be a double stranded also. So these are the ligand which we use for in vitro study, like a 5' triphosphate, and this is the virus which we use for in vitro experiment in the lab. But these two sensors can sense a variety of viruses and cause the pathogenesis. Okay? Poly-IC is basically a substance which it's like a double-stranded RNA. When we introduce inside the cell, then it will act as a virus infection. So without using virus, we use the poly-IC. So again, this is a signaling in a simpler way. There is an adapter known as IPS1. This adapter is recruited to the RIG-I and MDF-I, and then that upon recruitment, this will activate this whole complex, will activate this kinases, and then there will be a synthesis of type I interferon and pro-inflammatory. So this is about the RLR. I am giving you very short this thing. Now I will talk about the NOD-like receptor. So there are two major group of NOD-like receptor. One is which is present inside the cytoplasm, and they sense the variety of ligand. Here you can see a variety of ligands are there. So this NOD1 and NOD2, basically they sense and then this activate the NF-kappa B and MAP kinases pathway and induces the inflammatory cytokine. There is another group of molecule or NOD-like receptor family members which makes a multi-protein complex. And this multi-protein complex, we call it as an inflammasome. And these inflammasome basically activate the caspase 1 and caspase 11 in okay. And once this caspase get activated, then this will cleave the inactive form of pro-inflammatory cytokine known as IL-1 family cytokine. And IL-1 family cytokine is consist of three major cytokines. One is IL-1 beta, IL-18, and IL-33. Okay, and this is very potent cytokine. And try to understand this since this is quite potent inflammatory cytokine. There is an additional regulation by the inflammasome. Okay, so this is a simple signaling pathway for NOD1 and NOD2. And this is a multi-protein complex. This I was talking multi-protein complex, and this will basically activate the pro-IL-1 family cytokine to the active IL-1 form of cytokine, and this will be secreted, and then that will induce the appropriate response. With that, I will stop here, and thank you. And sorry for, I think I have taken a little more. I'm happy to take any question if you have.
Yeah. Thank you, Dr. Himanshu. That was a good talk, giving us an overview about the basic innate responses. So I would like to open the session for the questions from our team members.
Yeah, sure.
Yeah, I think I'm Dr. Chandrasekhar. Given the specification, in any of these autoimmune disease or any of the autoinflammatory disease, is there any specific reason why these proportions of this TLR and other recognition particulate ligand, does it alter or does it change?
Yes. So in autoimmune disease, there is some kind of dysregulation of inflammatory cytokine, and it could be, you can understand, it could be at any level. It could be at signaling level. It could be at, for example, if the negative regulators are hampered. For example, there are so many negative regulators. Okay, so if this negative regulator has some mutation, then even a small amount of ligand can trigger the auto-inflammatory condition. For example, if you take the case of SLE, okay, so SLE, in case of SLE, so cancer and autoimmune disease is a very complex. I cannot say there is only one factor which results, it's a multifactorial. So unfortunately, those individuals, they have a more than one factor, and that results to the development of autoimmune disease. We have recently shown that there is some microRNA. This is our recent work which we have published. So this microRNA basically binds to the negative regulator of TLR signalings. Okay, there are so many negative regulators, and in case of SLE patient, the expression of these negative this microRNA is very less. So when there will be a it's the other way around. The expression of microRNA is too much, and then this negative regulator is not there. So that will keep on switch on the TLR mediated signaling, RLR mediated signaling, and so on and so forth. So this is a one thing. And if there is some PAMP which is for example, in normal individual, this can be easily taken care by the signaling and cleared by the PRRs. Okay, but in some cases this cannot be cleared, and then this will activate the system, and then I explained you there is some kind of positive feedback loop. Okay, then we need to damp it by, for example, we give the steroids and all those things. Those patients, they receive a steroid and all those things.
Thank you.
So Himanshu, this is Girdhari.
Yeah.
Fantastic, very nice talk, and nice introduction of the system, innate immune system, and different molecules, how they work, and what are different signaling molecules. So my question, just for sake of the audience to get more details in that, what we explained to you is that majority of them are the ligand coming from outside source. So Himanshu, if you can explain what are the component in terms of which are indigenous source, which activate these ligands, some light on that.
So I did not go, I should put one more arrow. I showed the PAMPs, okay, yeah. Pathogen associated molecular pattern, okay. I should put one more arrow, but it will take more time. We call it as a DAM, danger or damage associated molecular pattern. So these danger and damage associated molecular pattern are endogenous molecule. For example, the DNA molecule, if you if somehow this DNA molecule is sensed by the innate immune cell, then it will induce the inflammation. And if there is a very good, beautiful study of Tasco Honjo. He worked with DNAs. He showed that if you mutate the DNase enzyme, which basically clears the damaged DNA, then that mice will develop the autoimmune disease. Okay, so there are so many, all molecules which is hidden inside the cell, they are potential dam. That includes DNA, that includes HSP. HSP is a very potent inducer of this TLR. Okay, so all those molecules which is hidden and present inside the cell, they are weak. And if by some way they are open up and they came out, then that will induce sufficient or quite a strong amount of inflammation. And that inflammation, we call it as a sterile inflammation. A very good, and generally I tell in the class, if you have a sterile hand and you put it in the clean bench and you make a cut, you will have all signature of inflammation, right? Over there, there is a no microbe, no pathogen. Okay. Or you take the say, just take the PBMC and take the PBMC and take one portion of PBMC and induce the hypotonic shock. So the cells will damage. Okay. And then you put same lysate or same thing in the healthy cell, you will see very beautiful production of IL-6, TNF, or whatever you want to say. COX, you can see the expression of COX and all those things. So this is about the sterile inflammation. I think I have answered, right, Girdhari?
Thank you. Yeah, perfect. No, thank you very much.
Yeah, this is Mohan Wani. I had just one quick question. Dr. Himanshu, it's very nice talk.
Thank you.
I have one small question. Is any specific TLRs are identified in COVID infection?
Yeah, it's a very beautiful question, cute question, I will say. So in COVID infection, mainly, of course, I don't know, but mainly the molecule which is involved is this RLRs. Okay. So, you know, COVID is or SARS-CoV-2 is a RNA virus. Okay. And this RNA virus is, they will transport the RNA molecule inside the cell. And over there, we have a RLRs, the sensor of RNA molecule. And these molecules are sensed by the RIGI, MDA5, even this will, so this senses, and TLR also probably, I don't know precisely whether most likely TLR8 is sensing over there. And this TLR8 senses and induces the inflammation, okay? But mainly RLRs are playing very important role in sensing this RNA viruses.
Okay. Thank you, Dr. Himanshu.
And please remember this inflammasome also, especially NLRP3 inflammasome, they also sense the RNA molecule, and that will produce highly potent this IL-1 beta or IL-18. You know, IL-18 and IL-1 beta is just like a small bomb, okay? And that induces, again, second wave of inflammation like CRP and all those things. And then all these things. So in case of COVID, there is a multi-level of inflammation. Virus is causing severe cell death, and that dead cells also inducing inflammation. And overall, that caused the cytokine storm kind of situation.
Okay, thank you.
Yeah,
I'm done.
All right. Thank you, Dr. Himanshu, for that wonderful talk. And